Lipid transport proteins, otherwise known as LTPs, are essential components of the intricate process of lipid metabolism. This is because they ensure the seamless transport of lipids across various cellular compartments. They also play a vital role in lipid homeostasis, which is a big part of cellular function. This comprehensive overview would delve into the classification, limitation, structure, function, and the historic event that led to the discovery of these LTPs.
To move to the classification and functions of lipid transport proteins. Under these classifications, we have the lipoproteins and the apolipoproteins. So under the lipoproteins, we have the chylomicrons, we have the VLDL, which is very low density lipoproteins, we have the LDL, which is low density lipoproteins, and the HDL, which is high density lipoproteins. Altogether, these classes of lipoproteins aid in the transport of lipids to the bloodstream. Next is the apolipoproteins. Under these classes of lipoproteins, we have the ApoA, ApoB, ApoC, and ApoE lipoproteins. So these lipoproteins help in lipid metabolism. Altogether, the physiological functions of lipid transport proteins include energy metabolism, membrane structure, and cellular signaling.
Structural diversity of LTPs. LTPs have diverse structures that determines their functions. And we have apolipoproteins. Apolipoproteins are large multi-domain glycoproteins. That means they are located outside of the plasma membrane. Then we have SRB1, scavenger receptor class B1, which are integral membrane protein. They are embedded within the plasma membrane. Then we have the FABPs. FABPs are fatty acid binding proteins that transport fatty acids, and they are cytoplasmic proteins as well. Then we have PLTPs. These are phospholipid transport proteins that are responsible for transporting phospholipids.
There are certain control mechanisms that have been put in place by the cell to control the amount of lipid transport proteins that are produced. As we all know, the cell always wants to maintain homeostasis. Now, considering the central dogma of molecular biology, we know the DNA is transcripted to mRNA and later translated to proteins. At these stages, the production of lipid transport proteins can be regulated. At the post-transcriptional stage, we can have RNA splicing and RNA decay. At the transcriptional stage, we could have the transcriptional factors that could turn on or turn off the genes. And at the translational stage, we could have modification of proteins. All this leads to homeostasis of lipids in the cell. Without homeostasis, there could be certain implications.
This regulation of lipid transport proteins is implicated in various diseases. ApoB, ApoAI, and SRB1 are linked to cardiovascular diseases such as atherosclerosis. FABPS, FATPS, and PLTP is linked to metabolic disorders such as obesity, diabetes, and non-alcoholic fatty liver diseases.
The discovery of LTPs evolved through key experiments spanning the 20th century. Early observation by Mashabuev in 1929 hinted at lipid-protein complexes, while ultracentrifugal studies in the 1940s to 1950s identified lipid-protein complexes. Glowat cell-free assays in 1963 and Lusky's identification of CETP in 1972 provided definitive evidence for specific lipid transfer activities.
In conclusion, LTPs are integral players
in cellular lipid metabolisms. They ensure the precise transport of lipids and contribute to various physiological processes. Understanding their structure, their function, and their implementations on diseases helps in developing targeted therapies for lipid-related disorders.